morning, students. So in this class, we will again discuss that is genetically engineered microorganisms. So in previous class, we have seen basic introduction to genes. So now we see the detailed procedure how we modify the genome of a bacteria. So genes is called as genetically engineered microorganisms. Genetically engineered microorganisms. So what is meant by this genetically engineered microorganisms? So in genetically we modify the genome. We modify the genome of microorganism so that it can attain special characteristic features which is not present when compared to other. So that is the importance of gene. So in genetically in microorganisms we will modify the genome. So why we use microorganisms to produce the foreign proteins? So there are two reasons. One is so the replication time of this of microbial genome is less, so we can get our desired product in less time. As the microbial organisms will replicate in less time or duplicate in less time, that is within 20 minutes. Within 20 minutes, the microbial cell will be duplicated. So in less time we get more protein, more gene product. And next one is environmentally friendly nature. Friendly nature of microbial organisms when compared to chemical synthesis of proteins. So we prepare the genetically engineered microorganisms by using our DNA technology. Or DNA technology is called as recombinant DNA technology. So where there are certain molecular tools to prepare the genes. So first one is gene transfer method. So in gene transfer method, we have to transfer the foreign gene insert into the microbial host genome. So we have two methods. One is electroporation and conjugation. Gene transfer methods, so by which we can transfer the foreign gene insert into the host genome. That is gene transfer method. That is electroporation and conjugation. We have two methods, electroporation and conjugation, by which we can insert the foreign gene. So electroporation, so we'll apply certain electric voltage, so that the bacterial cell can uptake the DNA. Whereas in conjugation, this is by formation of conjugation tube, the DNA will be transferred from one cell to the other cell. So this is gene transfer method. Second one is cloning vectors. cloning vectors are the vehicles which carries the gene of interest, which includes plasmid, bacteriophage vectors, and yeast vectors. So the cloning vectors are there. We can use different cloning vectors for transfer of foreign gene insert into the bacterial cell. So if you take the bacterial genome, which is a circular DNA, so where we can insert our foreign gene. So the place where we insert the foreign gene is called as MCS, which is called as multiple cloning site. So this is the site at which we have to insert the foreign gene. That is our desired gene. So if we take the plasmid, so we have certain regions. So one is MCS, where we will insert the foreign sequence, that is foreign gene. So in plasmid there will be MCS. Next one is antibiotic resistant gene for selection, and third one is origin of replication, that is ORS. So we'll cut the plasmid with certain restriction enzymes. We can cut the plasmid with certain restriction enzymes. So we cut the plasmid with certain restriction enzyme at MCS, that is multiple cloning site, and we cut the insert with the same that is gene of interest. This is plasmid. So we'll cut the plasmid and gene of interest with the same restriction. Yeah? For example, if we take two enzymes, that is EcoR1 and second one is In3. So we have to use same restriction to cut the plasmid and gene of interest. So which produces same stickiness, the restriction digestion of this plasmid and gene of interest with the same enzyme, it produces similar stacks. We call it as similar ends. Similar ends will be produced when we cut the plasmid and gene of interest with same restriction enzymes. So after the restriction enzymes, we will add an enzyme that is ligase, enzyme ligase. So this ligase will ligate the plasmid and gene of interest. We ligate the gene of interest and plasmid to form a recombinant vector, recombinant plasmid. So the insertion of foreign gene into the plasmid will result in the formation of a recombinant vector. So this recombinant vector will produce the protein of interest. That is whatever the gene of interest will express. So this protein will be expressed. So whatever the recombinant plasmid will be inserted into the bacterial cell. So this is bacterial cell. So after formation of recombinant cell, so the recombinant cell will be introduced into the bacterial host system. So either by electroporation or by conjugation. So this is gene transfer method and cloning methods. The third one is promoters. So the gene of interest, whatever the gene of interest, should be tagged with certain promoters. So there are two types of promoters. One is constitutive promoter and inducible promoters. So these promoters will help the in expression of that gene of interest. So constitutive promoters is nothing but continuous. So the promoters which are used to for the expression of genes continuously. So whereas inducible promoter, which allows the expression of genes only in presence of inducer, that is inducible promoter. So we can use either of this promoter according to our product, that is promoters. Next, fourth one is selectable markers. Selectable markers. So the selectable markers include mostly the antibiotic resistant gene, antibiotic resistant genes, antibiotic resistant genes, which includes, for example, which includes kanamycin resistant gene, tetracycline resistant gene, tetracycline resistant. So these selectable markers can be used to differentiate the recombinants from non-recombinants. Recombinants from non-recombinants. Recombinants from non-recombinants. So these are the strategies employed in RNA making. So there, how we modify the genome, right? There are two strategies for modification of the genome. So one is removal or destruction of undesired genes. Next one is overexpression of genes, overexpression of gene. So these are the two strategies how we employ for the introduction of foreign gene into the host system. Foreign gene into the host system. So we have certain applications of this genetically engineered microorganisms. First, we take in human health. So applications. We take applications. First one is human health. So in human health, these modified organisms we use for the production of therapeutic proteins. Production of therapeutic proteins, which includes human insulin, human insulin, human growth hormone. So human insulin, human growth hormone, recombinant vector, recombinant vaccines, recombinant vaccines are used in human health. In textile industry, coming to textile industry, production of amylase, which removes starch content from fabrics, is an application in textile industry. And next one is bioremediation. Bioremediation. Bioremediation, which includes use of the microbial organism, that is Pseudomonas putridea. So this is bioremediation. So it is an important application of genetically modified organisms. Pseudomonas putridea is microorganisms into which, so there will be lot of genes has been inserted. One is opt gene which metabolize, which break down the obtains cam gene, which break down the camphor, NAM gene, which breaks down the naphthalene. So the introduction of any of the genes in Pseudomonas putridea will help in the bioremediation. So opt gene, which is used for the breakdown of oil spills, breakdown of oil spills, breakdown of oil spills. So this is an important application of gems. Next, in food industry, after bioremediation, in agricultural industry. So introduction of new genes, that is nitrogen, introduction of nitrogen fixation genes into the genome will help in the nitrogen fixation. And coming to the food industry, so cheese production. In cheese production, the bacteria, that is Endothea parasitica. Endothea parasitica is an microorganism which is used for the cheese production, So this is all about the applications and how we prepare the gems. So we will once again come with the gems. So gems are called as genetically more engineered microorganisms. So where we modify the genome of the microorganisms by introduction of foreign gene insert. So as the duplication time of these microorganisms is less, so within less time we can get more amount of protein. So we use microbial organisms. And second reason is they are environmentally friendly when compared to the chemical synthesis of the same protein. So based upon these two reasons, we choose the genetically modified microbial organisms. So by using rDNA technology and the tools of rDNA technology, we can modify the genome of microorganisms. One is the molecular tools, which includes first one is the type of gene transfer method, so which may be done by electroporation and conjugation, where we insert the foreign gene sequence into the host genome. So this foreign gene insert is carried by vector, by a cloning vehicle that is called, we have different cloning vectors, that is plasmids, bacteriophage and yeast vectors, based upon the size of insert that we are going to insert in the host gene. Coming to the promoters, so there should be a promoter to enhance the increase of expression of foreign gene, that is promoter, constitutive promoter and invisible promoter, which includes continuously constitutive promoter which continuously expresses the protein, where invisible protein which induces only in presence of inducer. Next, selectable marker. Selectable marker so mostly which are used for the antibiotic resistant, mostly used antibiotic resistant genes. So that is kanamycin and tetracycline. So these antibiotic genes or selectable markers which are used to which are used to differentiate the recombinants from non-recombinants. So if you want to modify the genome, so the plasmid, we have to select the plasmid and we have to insert the genome, gene of interest, and this plasmid is transferred into the host. So if you take the plasmid, say which contains a different site that is called as multiple clone site, where we insert our foreign gene insert into the MCS. So after MCS, so we use the same restriction enzymes to cut the plasmid and desired vector, desired gene of interest. After restriction digestion, we will ligate the vector and gene of interest, which results in the formation of recombinant plasmid. So this recombinant plasmid is introduced into the host bacterial cell either by using electroporation or conjugation. So the steps involved in the generation of genetically engineered microorganisms. Coming to the applications. So mostly coming to the human health. In case of human health, it is used for genes are used for the production of therapeutic proteins, which includes human insulin, human growth hormone, recombinant vaccines can be produced by using gems. In textile industry, amino acids can be produced to remove the starch from fabrics. And coming to the next application, that is bioremediation, that is degradation of removal of waste by using Pseudomonas proteria, where we insert special genes into the host genome. So if we use octgene, which breaks down the octane of oil spills, CAMgene, which can degrade the camphor, NADgenes, which can degrade the naphthalene genes, which can break down the naphthalene. And coming to the agriculture, introduction of nick genes, which are responsible for the nitrogen fixation, And in food production, food industry, Endotheca parasitica is a microbial organism which is used for cheese production. So this is all about the genetically engineered microbial organisms. So with this we have completed genes.